We did not spend too much time in understanding viral identification because it was just not an easy task, and I don't want to go too much into discussing how to do those. However, viral multiplication is important to understand, and we do want to understand the process of viral identification. That is a nucleic acid that contains in a virion contains only a few of the genes that are needed for the synthesis of a new virus, and these genes will include basically the structural component, which is the genetic material, and the capsid proteins. And the process of viral multiplication is important for us to understand how easy a virus can enter a host cell and take over the host metabolic machinery. A single virion can sometimes give rise to several thousands of similar tiny viruses in a single host cell, and this process can drastically change the host cell and cause disease, and sometimes it may even lead to a fatal condition. The multiplication of viruses can be explained by a simple growth curve, in which we can see that usually once infection has entered the host cell, it doesn't take too long for the virions to release from the host cell and cause an acute infection. So how do viruses multiply? As I said, for a virus to take over the host's metabolism is the first thing that it would need to do, is because viral enzymes are almost entirely virus does not have its own enzymes. It's actually using and synthesizing the function only within the host cell. So it takes over the machinery of the host cells. So it will use the host cells, proteins that are manufactured through the ribosomes and the tRNA of the host cell. All its function is now controlled by the viruses. So, although by means a virus will enter and exit a host cell by simple means, but we will try to understand that based on two different ways of multiplying, and these two alternative mechanisms are called as the lytic cycle or the lysogenic cycle. The lytic cycle ends with the death of the cell of the host and the lysis of the cell, which was that the cell burst, whereas the host cells the lytic cycle will end with the lysis and the death of the host cell, whereas the host cells remain alive in the lysogenic cycle. And we will look at both life cycles with T-even bacteriophages that are the ones that are studied the most, and most of these infections have been in E. coli. So T3 and T4 are mainly kinds of bacteriophages that are large and complex, and they're actually non-enveloped. They have the characteristic of the typical space shape type. That is, they have a head and a tail that is shown to you in this picture up here. The length of a DNA in these viruses is about 6% of that contained in the E. coli, and thus the phage has enough DNA for over 100 genes. The multiplication of these viruses has 5 major steps. The first step is called as attachment. That is a chance for a collision between the phage particle and the bacteria. Attachment is done through adsorption, which means that the virus attaches to the complex receptors on this bacterial cell, and it will penetrate through the tail fibers. This attachment is mainly a chemical interaction in which weak bonds are formed between the attachment and the receptor sites. Once the attachment process has passed on, the next step is penetration. That is, the T-even bacteriophages will inject its DNA or the nucleic acid into the bacteriophages. As I said, it attaches up here with the tail, and then it injects in there. So think of a syringe. You're actually injecting the inside, which is the nucleic material, into the whole cell. To do this, the bacteriophage will actually release enzyme or the lysozyme, which is released through the tail sheath of the phage. And once it does that, the DNA from the bacteria's head region, which is the region up here, will actually pass through that tunnel and enter the inside of the bacteriophage, that is the host cell. Now, within the host cell, once the bacteriophages has reached the cytoplasm of the host cell, the biosynthesis of viral nucleic acid and protein starts taking place. So here is all the little, you can see the little heads and the tails are being formed in here. It takes the host protein synthesis is stopped by the virus degradation of the host DNA, and the viral proteins interfere with transcription and or the repression of translation of the host cells. Initially, the phage does use the cell's nucleotides and several of its enzymes to make copies of its own phage DNA, but soon the biosynthesis of the viral proteins will become, and any transcription that is going on will be from the phage DNA to the mRNA to its proteins, and its proteins are mainly the capsid, which are used for the protein coat. So the host cells, ribozymes, enzymes, and amino acids are all used for by the phage. And after this biosynthesis is actually a long process, and after several interval of minutes have passed, the phage it actually starts going through ineffective stage in which it will the viral multiplication is actually almost complete, so it goes to a stagnant stage, and soon after that it goes to the next stage, which is called as maturation. In the maturation stage, the bacteriophages and the capsules are now assembled into little tiny virions, and these there are tons of these virions are forming within that maturation phase. That is, the subunits and the head are put together. And finally, we follow the last stage, which is called as a release state. In this cell, the host cell actually will lyse. Actually, I went too fast, so we skipped that. Got there. I'm sorry, my slide went away. So the host cell will actually lyse, or that is burst, and the new virions are released. And the term lysis is generally used for the stage in this multiplication of the T-even phages, because in this case, the plasma membrane actually breaks open or lyses, and that's why it's called as the lytic cycle. So let's go to the next couple of slides. So here's the main question is, what is the difference between a lytic and the lysogenic cycle? Now remember, the lytic cell will cause the lysis and death of the host cell. The lysogenic stage, on the other hand, that we will look in the next diagram, is the one in which the prophage of the DNA is incorporated into the host DNA. And that might ring a bell of something we did earlier in the microbial genetics chapter. We talked a little bit about transduction, in which the phage enters the host cells and becomes it takes over the genetic material of the host and actually incorporates into the host DNA. That's the right term to use. And this is the lysogenic phase. So up here you can see on the left is the same cycle about the lytic cycle, how the phage DNA is infected. Now once the phage DNA is infected, it actually has two options to do. It can either go to the lytic cycle, which we just finished discussing, or it can go to the next one, which is called the lysogenic cycle. In the lysogenic cycle, you can see the phage has actually incorporated their DNA into the host cell's DNA and begins the lysogenic phase. In lysogeny, the phage remains latent or actually inactive, and the participating bacterial host cells are now known as the lysogenic cells. And we will use the example of bacteriophage lambda, a very well studied lysogenic cycle in the E. coli. So here is the example in much more depth. Now notice in the first stage, it is the prophage. It will exist in the galactose using host that is shown up here. It's penetrating the E. coli, and within the E. coli it actually penetrates and I'm actually going to show you up here in the first one. No, we don't see it over here. No, we do see it over here. It actually enters in here and splits open and enters the host DNA. So it becomes part of that the DNA that's present in a circle. Once it's in there, the phage will continue. It will take over the genome and will now multiply along with the replication of the host DNA. And eventually the phage will mature and it will split open from the cell, releasing a phage that will carry that particular gene within it. The phage will then infect a host cell that, for example, in this particular example, they've discussed the example of galactose. Now, once the phage infects a new cell that cannot utilize galactose or lacking that gene, the galactose this cell is a galactose negative recipient cell, and along with the prophage, the bacterial galactose gene becomes integrated into the new host cells, and now this new cell, the same cell, can now metabolize galactose. Earlier, the cell was not able to break down galactose. So what it's doing is it's infecting one cell and taking the gene of galactose metabolism from the first bacterial cell and entering a second bacterial cell that was negative for galactose and converting it or changing it, or we can call it the genetic modification to the host cell so that it's able to break down galactose. Now this is something we did earlier. This is very similar to transduction that we talked about in microbial genetics. So how do viruses multiply in animals like us? So here is the same terms that we did before. Attachment. We have attachment, penetration, the uncoating by the viral or the host enzyme, biosynthesis, that is the production of the nucleic acids, maturation, and eventually releasing of the virus. So this is pretty much a repeat of the slide that we did earlier. What's different about entry? Let's look at it in more closer depth. Attachments for both will depend on person to person. It's mainly the receptors that's the cell looking at. And notice during the penetration process, you can see how it's forming the pinocytosis or the cell drinking, as it was called in biology. So that is an active cellular process, which means that it's an active process, requires ATP, in which the nutrients and other molecules are brought into a cell. So similar to that, here is a virus being taken in towards the membrane that is folding inwards to form this vesicle. And these vesicles contain elements that originate from the outside and are brought inside the cell. Once it's inside the envelope, the virus can then undergo a process called as fusion, in which the viral envelope will fuse with the plasma membrane and release the capsid inside. That's pretty much done, for example, in HIV. That's how HIV penetrates the cell by this method. The next step is called as uncoating, which is a virus will disappear during the eclipse phase of an infection because they're taken apart inside the cell. That is, they are it's uncoating, which is a separation of the virus nucleic acid and its protein coat, which is inside the host cell. And the capsid is actually digested when the cell attempts to digest the vesicle. And after the enzymatic degradation of the protein, the viral DNA is going to be soon synthesized, and that's when the infection, actually we say it's beginning. And when a viral infection is going on, when the virus is multiplying, that's when, for example, in us, when we see the temperatures rising after a flu or a cold, it's because the virus is multiplying within the cell. So a high fever is usually a sign of a viral multiplication. The final stages, as I said, maturation, in which the nucleic acid and the capsid will be put together. It's mainly the assembly that's going on at this stage, and release of the envelope is done through a process that is called similar to budding, in which the envelope actually develops around the capsid by a process called as budding. You can see up here the viral capsule and the host cell membranes forming these little buds, and eventually releasing them and being released to the outside. Here is a slide that I just put it in just to give you a vision of multiplication of a DNA virus. This picture is also one in your book. So again, from step 1, which is the virion attaches to the host cell, to the release, all the steps that we just discussed are in here. Here's an alternative pathway of multiplication used by many RNA-containing viruses. RNA-containing viruses usually, for example, in the first step up there, you can see the uncoding of the single strand of the RNA virus, and sometimes you have a positive and a negative strand. That is, a positive strand is shown to you as one from one cell, and the negative strand from the other cell. So I think I'm going to skip this and not go too much into it, as long as we understand the lytic cycle and the lysogenic cycle and the maturation phases. I'm going to stick with those. So we're going to skip these two slides, just sort of know their images and not much into it. I will try to show to you in class the multiplication of a retrovirus, and the retrovirus is actually very interesting, and we want to understand what a retrovirus does. A retrovirus is a virus that actually transforms from once a retrovirus enters a host cell, it actually is an RNA virus, and it actually incorporates to form a DNA. And that viral DNA is then transcribed or actually becomes a part of the host cell. And once it becomes a part of the host cell, the new viral DNA is transported from the host cell's nucleus to its central dogma and start making its proteins. Now, these proteins that are made because of the host cell's incorporation are no more they destroy the humans, they destroy the immunity. For example, the example that I can think of right now is the AIDS virus. What happens is that in the AIDS virus is HIV is a retrovirus in which it enters the host cells, and it's coming its viral DNA, excuse me, it's making its viral DNA, and this viral DNA becomes a part of the host cell. Now, during transcription of the previous cycle up here, new RNAs for these new retroviruses in the genomes and the RNA that encodes the retrovirus capsid and enzymes are all made in here. These viral proteins are sometimes processed by viral enzymes. Some of these viral proteins are moved by the host cell, and eventually mature retrovirus leaves the host cell, and it acquires an envelope, and it has spikes, and it can go and attach another cell. So a retrovirus is the opposite of a usual phase, and this is called as due to the process of reverse transcriptase. A reverse transcriptase is when a viral DNA infects into the nucleus, reversely makes copies the RNA, mRNA to make the DNA. Remember, DNA makes mRNA and mRNA makes proteins. So this is the opposite of that. So we are going to stop over here and then continue with the remaining of this lecture later.